Welcome to the Marineff Breakwater Block Monitoring video. My name is Jess Bone and I'm a Research Assistant for the Marineff project. This video will give you a more detailed overview of the monitoring protocols created by project partner, the University of Caen, Normandy, via the Borea Laboratory and the CREC Marine Station. The Marineff breakwater blocks have been designed to promote the colonisation of benthic organisms compared to the breakwater blocks already existing in the marine industry. They have been modified while retaining the mechanical characteristics of their primary function. Cavities of different shapes and sizes have been added to the breakwater block to recreate microhabitats found in hard substrate marine ecosystems, such as rocky reefs. The surface of these breakwater blocks meets the eco-engineering design criteria that we define here as experimental and has been modified to make it rougher and favour the attachment of benthic organisms, while the surface of the plain breakwater blocks remains smooth. 4 rings have been added to the top of each breakwater block in order to be able to fix the benthic chamber that forms part of the breakwater block monitoring protocol. To address the research questions of the Marineff project, in particular on the difference in colonisation between plain breakwater blocks and experimental breakwater blocks, both types of breakwater blocks were immersed. Both types are present at each site, including Cherbourg Harbour and Bernier-sur-Mer in the Bay of Seine, with 6 of each type of block in order to have 3 replicates of each. Therefore, in the Cherbourg Harbour, 12 breakwater blocks were immersed subtidally and 12 in the intertidal zone. Another 12 breakwater blocks were immersed subtidally in the Bay of Seine at Bernier-sur-Mer. We therefore have a total of 36 breakwater blocks for environmental monitoring, with 18 plain and 18 experimental. The 2 deployment sites chosen for these experiments have different environmental characteristics. The first site, in the Bay of Seine, is located off the coast of Bernier-sur-Mer. It is an environment exposed to strong hydrodynamics with a rocky flat bottom with some sandy areas. The second site is located next to the large promenade of Cherbourg inside the eastern breakwater. It is an area sheltered from hydrodynamics on a sandy seabed. Both subtidal sites are about 10 metres deep at high tide and 4 to 5 metres deep at low tide. The objectives of environmental monitoring are to quantify the primary production and productivity of algae, including both micro and macroalgae, as well as the diversity, abundance and biomass of the different trophic groups of benthic fauna present on the breakwater blocks. The primary production and productivity measurements are carried out using an automated benthic chamber developed by the Borea Laboratory and the CREC Marine Station, which is shown in the photo here, and using a diving PAM, which is a submersible fluorometer. For benthic fauna, quadrat photos and scrapings are made to determine diversity, abundance, and biomass estimates. Environmental monitoring is carried out 4 times a year for each season over 2 years. 3 replicates are taken for each measurement. For each block, 3 identical measurements are made for each season, which improves the robustness of any data gathered. So for 3 replicate measurements made 4 times a year over 2 years, we need 24 different measurement surfaces to perform the environmental monitoring without measuring the same area twice. This allows us to follow each stage of the biological colonisation and succession of the breakwater blocks by benthic organisms. A rotation is therefore operated between the measuring areas, as demonstrated in this image. Here is a diagram showing the complete system for measuring primary production on an experimental breakwater block. This system was specially developed for the Marineff project. It consists of an opaque cylindrical enclosure and a cover with an LED panel. This LED panel is controlled and powered by the LED driver system, which allows us to program the light intensity and exposure time of each intensity, making the driver system autonomous. The system is fixed on the black ring seals on the top of the breakwater module, hermetically sealing it from the ambient conditions. The benthic chamber is then connected to a multi-parameter probe, which measures oxygen, temperature, pH, salinity and turbidity, and a PCO2 probe, which measures the partial pressure of CO2 on a closed circuit facilitated by a self-contained submersible pump. On this video clip, you can see the complete primary production measurement system in place on a breakwater block at the Cherbourg site. The sensors used allow us to obtain data on turbidity, salinity, temperature, pH, oxygen concentration, and CO2 partial pressure. Water samples during the measurements are analysed to gather data on the nutrient concentrations of the surrounding water body. Dissolved inorganic carbon measurements are also obtained from samples taken at the beginning and end of incubation. At the end of these oxygen measurements, calculations allow us to estimate the primary production per unit area and the primary productivity of macroalgae. Here is an example of the results that can be achieved. The curve on the left shows the so-called production energy curves obtained on plain and experimental breakwater blocks. The light intensity, presented on the x-axis and referred to as photosynthetically activated radiation, abbreviated to PAR, gradually increases within the benthic chamber, which leads to an increase in photosynthetic activity. This results in the increase in oxygen production, as shown on the y-axis of the graph, until reaching a plateau. This plateau corresponds to the maximum capacity of the algae sampled. The results of the graph on the left are from the growth on the surface of the breakwater blocks, while the graph on the right shows the productivity results from macroalgae. We therefore measure the production of the breakwater block surface and the productivity of macroalgae. This video clip demonstrates how scraping samples are taken once the primary production measurement is complete. Scraped organisms and algae are sucked up using an underwater vacuum, topped with a 500 micron mesh net to catch the sucked up fauna. A second measurement on photosynthetic organisms is carried out with a diving pan. This device makes it possible to measure the variation in fluorescence of algae according to flashes of light made in the dark and under different levels of light. This measure allows us to estimate the physiological state of algae and to have estimates of the maximum photosynthetic activity and capacity of the species measured. The measurements are carried out on macroalgae thalli, chosen randomly on the breakwater blocks and on microalgae present on the surface of the breakwater blocks. The microalgae growing on the breakwater blocks is sampled using the scraping method on a surface of 25 centimeters squared. The sample obtained from scraping is recovered using a 1.5 litre syringe at the end of the underwater vacuum seen earlier. These microalgae samples are then analysed with lab staff. The lab staff measures fluorescence variations like the diving pan and makes it possible to obtain several photosynthetic parameters and primary production measurements. Lab staff's technology and measurement principle, however, are different from the diving pan and allow for many more photosynthetic parameters at different light wavelengths. The measurements are carried out on samples of resuspended microphytobenthos. The graph on the left shows the production capacity of a sample of microalgae scraped from the breakwater blocks, and the one on the right shows its physiological state. On each vertical face of each breakwater block, quadrats of 25 by 25 centimeters are photographed during each dive survey. These photo quadrats are taken on the same dedicated space each time. These photo quadrats make it possible to monitor the colonization of the breakwater block faces by benthic organisms and to quantify by counting when possible, or by a percentage cover by different trophic groups. Scrapings are also carried out on the photo quadrat area to determine the biodiversity and biomass present. In addition to photo quadrats and scraping samples, counts of mobile and sessile macrobenthic species are carried out. For each breakwater vase, benthic organisms are counted and visually identified with the most accurate taxonomic resolution possible. In addition to the macrobenthic count, a count of the fish present on the surrounding area is also carried out. Thanks for watching. There are more videos in the Breakwater block series which are listed on screen. Be sure to also visit our social media and the Marineff project website for more resources.